All right. Thank you, everyone, for joining today's session of the Knowledge Portal Network webinar series. I'm going to step through some updates from the Knowledge Portal, and then I'll hand it off to Noelle Burt, and then we will have our guest speakers present at the end. So first up, we'll be attending ASHG in Denver, Colorado in November. So if you'll be attending, we have both an exhibitor booth and an ancillary event. The details are here. This ancillary event will be geared towards predicted models for genetic support and enhanced visualizations integrating single-cell data with human genetics data. So we'll have a little bit more detail for you in the upcoming months, but please put this on your calendar if you are going to be at the event. We'd love to have you participate. We'll also be hosting a community workshop on standards and infrastructure for predicted effector gene lists on September 16th and 17th. There's a limited availability, so please register soon. Here are the details. We also have a news item about this as well. So also, if you have any questions or concerns, please feel free to reach out to us regarding this. But we're very excited to be hosting this. Other upcoming things for the fall, we have a production release that will be set for October 2024, and it will include various features and updates. A few are listed here, but these are not all of the updates that we'll be presenting. So keep an eye out for the new features and visualizations. As part of the production release, we'll have some updates to some of our community portals, including the aging KP, the neurodegenerative disease KP, the lupus KP, and pigeon, which is going to be new for us as well. Another resource that we've built for the Common Fund data ecosystem, the Knowledge Center, as you can see here, we'll also have a production release in October, and we'll include the following. So one thing that we're excited about is being a single entry point for all of the Common Fund. So essentially through this, we're hoping to elevate awareness of key resources and help researchers address specific scientific questions. And you'll be able to have some differential expression workflows that we'll be able to work through with you. And we're thrilled to announce that our latest portal, the Mammalian Adipose Tissue Knowledge Portal, or MATKP, will be unveiled in the production release as well. So keep an eye out for this. Essentially, this resource is going to be open access and will aggregate data regarding human adipose tissue, its cell type, the genes active within it, and its relationship to human disease. We're also hiring, so if you're interested in joining our team, we have a role open for a scientific communication and curation manager. We'll have a news item on this as well if you want to see any details about the role itself, or if you know of anyone that you would like to recommend for this role, we'd be eager to hear thoughts and opinions on that as well. But we'd love to have you join our team. And lastly, before I hand it over to Noelle, here are our upcoming meetings. So for September 12th, that will be our next focus group session, and October 10th will be our next webinar series. So with that, those are our updates, and I'll pass it off to Noelle.
Excellent. Thank you, Mackenzie. Lots going on. I sometimes get surprised about the things that we're doing. It's pretty cool. All right. I'm going to switch my screen. Hopefully folks can see this. I'm just going to give a brief framing before we have our speakers take over for today. And just because I'm really excited about this, and I also like to contextualize all the work that's going on within the portal team and the broader ecosystem in the framework of the scientific presentation here. So, today we're really sort of focusing on our common metabolic disease knowledge portal, sort of our centerpiece, our oldest of our resources, and the one that really has really amazing community involvement and engagement. And we're focusing on this particularly because the traits and phenotypes that we're going to talk about today a little bit really circle around common metabolic disease. But, this resource really serves as very specific functionality. And I think that's important to highlight today, because what we really hope to enable with this resource is that a human geneticist can have their life made a little bit easier, right? If they are studying a given gene or trait, they normally, without the CMDKP, would have to go to the GWAS catalog, download all the summary statistics, be able to run a meta-analysis, go and run a set of annotation pipelines like in FUMA GWAS, search the literature, mine model organism databases, understand gene expression results, kind of pull all that together, run a bunch of analyses, and then finally at the end, you may indeed be able to have a list of target genes and whatnot, right? So that's a lot of work. Human geneticists are really good at this, but what if you don't want to do that? You want to make your life easier and you really want to know what genes are involved in the disease you study, or, you know, what role it has in human disease. You know, our resource really aims to make that simple, right? To present that to you in one sort of framework that is vetted by the community, right? But done as a flexible platform with methods that are encoded so that run at scale for you, and then presented in a way that you can navigate. And really that's our core functionality. But this is sort of driven by 3 competencies, right? Community driven. And what I mean by that is contributors are our bread and butter. They are the people that matter to us in terms of like how to navigate and grow the resource from in terms of our roadmap, in terms of what we're building. We make that a flexible platform so that we can obviously always course correct and hopefully give on-demand knowledge. And this is evidenced by the fact that, you know, over 50% of our data sets from human genetics have all been contributed directly working with a collaborator, like spending time with them, understanding their phenotype, their data, how they want it processed and QC'd, and how they need it represented. And that's really been part of what we've found is very important to us to build a resource that folks use, right? So that makes the resource also something that pivots over time. And I'm showing this more, not to demonstrate how many people use the resource, but more that like in every quarter, we're thinking about different types of things to do to serve a research community. And this comes in the form of data sets, different methods, different visualizations, or simply just different approaches to serve up information as stewards of human genetics data for the community. But to truly do that well, right, the community is very diverse. And I think it's a challenge when you say, you know, what's the user experience? What's the story? What's the journey? And I think a lot of resources struggle with that to really get that right. And we really do too, right? But one of the things that I think we do really well is we've served sort of the human genetics and the computational community quite well because of one simple thing. They are both contributors and consumers. They are the folks that use the resource once they've contributed data to it because they're invested, and they're also using the resource to help with their resource, to sort of minimize the gaps and the pain points in going to gather the data themselves, run the analyses themselves, and view the results themselves. And so with that, you know, we also think about what are the other users. And when I think about our portal, like the best sort of complement really, or the best sort of expression of how we develop the resource going further is really someone who wants to help you improve your resource. Because that means A, they're using it, and B, they found a space where they think it could actually serve the community better or help them address scientific questions. And so today we're going to hear from that community represented by Attie and Mark Keller. And this actually started back in 2019 or 18, I think, at a workshop at the NIDDK. And we just gave a presentation of the portal. It was the early days of the portal. And I think I remember this pretty correctly. Alan raised his hand and says, hey, have you guys thought about mouse data and mouse genetics? It's really important as a model system for diabetes and related treats. And we're like, it's a great idea. Flash forward, you know, we had to serve the genetics community. We had a lot of things on our plate, but Alan came back, and I appreciate that, with Mark saying, you know, this is a story we want to tell because we want to use our data to help improve the resource and also share how we use it so you understand how to contextualize that for our community. So with that today, the mouse takes center stage, and we think about the future where we're not just a portal that represents human genetics data. Really, we want to represent mouse genetics data because that helps you understand what you might want to do experimentally, or the phenotypes, or what tissue, or all the scientific questions that you really have about your given gene or trait can be answered with more depth if you understand what's going on in the mouse. So with that set up, I'm going to turn it over to Alan and Mark to give our presentation today.
Kenzie, can you let me share? Oh, hold on. Now I can. Yep.
And then Mark, if you're okay with this, we'll let people raise their hands and pop in the chat, but Mackenzie and I will monitor it. So if there's anything that's sort of urgent, we'll let you know so we can stop and answer it. Is that okay with you as you go through?
Of course.
All right, fabulous.
Okay, so I'm going to give a few introductory remarks, and then I'm actually going to pass it off to Mark, who will give the majority of the talk. So thank you for allowing us to give this presentation. Can I have the next slide, Mark? So I'm going to tell you a little bit about the kinds of projects that we've done using mouse genetics. There'll be 3 that I'll talk about quite briefly. But the main topic is really going to be systems biology, how we take data from various sources and take advantage of genetics and correlation across Mendelian randomization populations and make discoveries. Next slide. So the first study I'll mention is one we did quite a few years ago that really addresses a core question in diabetes, which is the dichotomy wherein most people who have type 2 diabetes are obese, but most people who are obese are not diabetic. And so we're really interested in discovering what are the genes that make the difference in the context of obesity between diabetes susceptibility and diabetes resistance. Next slide. So we developed a model system, which is 2 mouse strains, B6, the commonly used B6 mouse strain, and BTBR, where if you introgress the OB mutation, leptin deficiency, and make them obese, you get 2 very, very different outcomes. And you can see by the red curves that the BTBR mice, both females and males a little bit more severe, are very diabetic, and whereas the B6 mice have a transient and modest hyperglycemia. Can you click? So with this model system, we did a lot of genetics and identified a whole bunch of genes in 2 major categories, ones that were involved in insulin production and secretion, and others that were involved in the regulation of beta cell mass. Next. So our earliest venture into propagating data, sharing data, was a website that we rolled out way back in 2008, but has turned out to be very widely used in the diabetes field. It's a very simple website, but it is incredibly valuable because it has really high quality, and at that time cutting edge, because it was the Illumina microarrays, where we looked at gene expression in 6 different tissues from both sets of mice, lean and obese, and then at the ages that preceded the onset of diabetes and then after the onset of diabetes, so that one could look at genes that were causal and genes that were altered as a consequence of diabetes. Next slide. And just to give you one little example of something rather dramatic that ended up resulting in a very nice discovery, you can see the effect of obesity on the expression of a gene called FOXM1, a transcription factor expressed in beta cells, where it was substantially increased by obesity in the B6 mice, but not at all increased by obesity in the BTBR mice. And this correlated very strongly with a direct measure of beta cell proliferation that we had and that we published. And so we were able to show that this gene was a very important one in insulin secretion. But along the way, we did correlation analysis, next slide. And you can see that this data is incredibly obvious, where there's strong correlation. The correlation coefficients are 0.98, and every single gene on this list is involved in cell cycle control. And so that was just a really nice demonstration that stimulation of beta cell proliferation was a key component of the susceptibility or lack thereof to diabetes in these animal models. Next. So there are problems with intercrosses. The genetic space you're exploring isn't that broad, and the number of meioses is very small, so you don't have a lot of mapping resolution. And this is a problem that Gary Churchill addressed by developing the diversity outbred mice. Very briefly, what Gary did was he chose 8 mouse strains that capture most of the genetic diversity of the inbred mouse strains, mainly because of the existence of the wild-derived strains, 3 wild-derived strains, CAST, PWK, and WSB, but then also incorporating the most widely used mus musculus inbred mouse strains, B6 obviously, AJ, NOD, a model of type 1 diabetes, NZO, a model of obesity, and AJ, which is very resistant to obesity and a lot of metabolic disease. So these mice are being maintained as an outbred stock at the Jackson lab. Next. And just to give you a very quick overview of the variation, the phenotypic variation, these are glucose tolerance curves, and you can see that some of the mice have impaired glucose tolerance because they're not producing enough insulin, and some of them have impaired glucose tolerance and have a lot more than normal insulin, like the B6 mice, and so are insulin resistant. So there's a wide phenotypic range, and quite honestly, we've looked at hundreds and hundreds of phenotypes, and any phenotype we look at has great variability across these mouse strains. Next slide. And so we looked at insulin secretion and saw a lot of variability. That's shown here in this heat plot. Next slide. I'm going quickly because we have less time than we expected. And I think I'm going to skip this slide because again, I want to spare Mark some time. So I'm going to skip this. It just shows you variability in insulin resistance phenotypes. Next slide. And then I just want to describe very quickly a study that we're in the middle of right now. That's a large study on the genetics of diet responsiveness. And the motivation for this study is on the next slide, which is that there's a large variation in diet response, and it makes it really impossible for people to recommend diets for individuals and to really know ahead of time. There are no biomarkers. There's nothing really hard, well-grounded in science that predicts what the best diet is for an individual. And here you see two individuals and their glucose responds to eating a banana or cookies. And they're exact opposites. The one on the top had a strong response to the banana, and the one on the bottom had a strong response to the cookies and not to the banana. And these are things that are not predictable. So we want to explore the genetic basis for the variation in diet outcome. Next slide. And so our study is on the DO mice, where we are measuring metabolic flux. We're measuring a lot of intermediate traits through RNA-seq, proteomics, lipidomics, and the microbiome. And we're integrating all this data to try to discover the genetics, the gene drivers, and the pathways downstream of the gene drivers that are responsible for difference in diet outcome. But it's generating a tremendous amount of exciting data that can be used to discover just general control mechanisms and taking advantage of these perturbations. Okay, over to you, Mark.
Thank you, Alan. So I want to pause for a moment, and Alan introduced these 8 strains that Gary Churchill and colleagues selected several years ago. And again, the level of genetic diversity contained within these mice is comparable to the level of genetic diversity contained within humans. And that was by design. And a lot of that is brought in by these 3 wild-derived strains, as Alan said. But also these other mice here bring in well-established models of various metabolic diseases. And so what I want to do is tell you a few stories for what we've been doing to evaluate the influence of genetic background in these mice, and then integrate that findings with what has been observed in the human, and then do the reverse. Start in the human population for genes associated with disease, and then go to the mice and see how does that inform us for which mice might be good model organisms to study that particular gene function, and go back and forth between these 2 organisms. And while mice are not humans, let me acknowledge that, I hope to convince you that the regulatory pathways that underlie disease risk are discoverable in the mice, and they offer excellent model systems to study them. So the 2 stories that I want to tell you about are really looking at gene expression. And for years, Alan and I have been integrating our mouse observations with the knowledge portal. And we're probably one of the biggest users of that. So I really appreciated Noel talking about, bringing more people into the community with the mouse data. And I want to show you in this first study, which was done in collaboration with Gary Churchill at the Jackson Labs, how strain background within these mice exerts a powerful influence on gene expression in these 5 major tissues, the islet being the source for insulin, and these other tissues being insulin target tissues, and how we could use the influence of genetic background on gene expression changes to inform us about what might be happening in the human. And then I'm going to also talk about, as Alan just described a moment ago, we've extended study number 1 by looking at the differences in 2 different diets. So study number 1, all of the mice were on a Western diet, high in fat and carbohydrate, whereas in study number 2, we repeated our survey of gene expression in liver when we put the mice on either a high fat or a high carb, and that is allowing us to start talking about the diet responsivity as a function of genetic background. So for the first study, we did RNA sequencing on these 5 different tissues. And what I'm showing you here is Charles Opara, who was a student in the lab, computed an ANOVA in each of these tissues to identify which transcripts were influenced by strain background, sex, because we had both sexes of all of those 8 mice, or by sex by strain interactions. And I'm going to show you an example of this as well. But I think you can appreciate in these histograms, which is plotting the number of transcripts and an adjusted p-value for whether strain is having an influence, is that islets by far showed the greatest number of transcripts that had a difference as a function of strain background. And that's really interesting to us. There were hundreds of genes, if not thousands, that were different as a function of strain background, but islets had the greatest number. Whereas for sex, while it did not have the magnitude of effect that strain did, sex appears to have a stronger influence in adipose tissue, shown in blue, or liver, than in islet. So that's very interesting to us, and we want to take advantage of that to start asking questions about, well, what do these genes look like for their human homologue in humans? And that's where I'm going. Just to give you a flavor of some of these genes that show dramatic influence as a function of strain, I'm plotting 6 here. And my purpose here isn't really to drill into a specific gene, but to illustrate that there are hundreds, if not thousands, of genes that showed differences as a function of genetic background. And each plot here is looking at the expression of a gene, the log2 value of that, as a function of strain background, which are listed across the top here and are color-coded. So for example, this particular gene has very high expression in AJ and NOD and relatively low expression in the other strains, whereas this gene has sky-high expression only in PWK. That same kind of thing is observed in every single tissue. So here are 3 genes in the liver, and once again, you can see dramatic differences as a function of strain background. In the bottom, I'm showing you 3 additional genes in adipose tissue. And finally, the skeletal muscle and the heart, again, show thousands of genes that are differentially regulated as a function of strain background. So what do we do with that information? The first thing we wanted to ask is, well, if there's genetic dependent regulation in these genes in the mice, is the human homologue of those genes associated with disease in humans? So we're going to start in the mouse and then identify those genes and ask, are they associated with disease risk in GWAS? And I'm going to introduce a relatively new metric developed by Jason Flanick, which is the Human Genetic Evidence Score, which, as I understand it, it factors rare and common genetic variants to give us a better sort of gene-centric view for that gene to be associated with a particular trait. And I'm going to show you one example, this gene called paraoxynase 2 or PON2, where in every single tissue, here I'm showing you islet, liver, adipose, muscle, and heart, the expression of PON2, as you can see, is dramatically lower in cast mice than it is for all other strains. Now, in some tissues, the PWK, which is shown here, is intermediate between the low expression in cast and the high expression in WSB. And one particular tissue actually shows a very strong sex effect. So not only do we have a strain effect for a difference in PON2 expression in every tissue that we've surveyed, we have a strain by sex influence as a function of tissue only observed in adipose. So that's very interesting. And the reason I'm showing you this particular gene is when I go to human GWAS and use Jason's score for genetic evidence, it turns out that PON2 is strongly associated with fasting insulin and HDL cholesterol. And that's just one example. And I was doing this manually, and then I worked with Jason to do it in a more systematic way, where he performed a multi-tissue regression to ask if genes that have a strong influence of strain background in our mouse study show enrichment for phenotypes in human GWAS. And what I'm showing you here in each of these panels is the result of that regression. And it's p-value adjusted, so I'm only showing you significant human phenotypes that are associated with genes in the mice that have a strong influence of genetic background. And again, the story that I'm developing here is that genetic differences in the mice that result in a change in the expression of a gene have a human homologue that are enriched with SNPs associated with various phenotypes. And what was interesting to us is there was a difference as a function of tissue. So for example, islet had many, many more human phenotypes associated with genes that were differentially expressed in the mice than, for example, muscle. And that's interesting to us. And it suggests that genetic regulation of genes in the islet are contributing to disease risk in humans. What are these phenotypes? And here I'm just showing you on the left those phenotypes that had the strongest association in the humans for genes that were influenced by genetic background in the islet. Many of them are associated with diabetes. And you would kind of expect that for islet genes to undergo dramatic differences in expression, might be related to diabetes-related traits. And I highlight a few of those in green. Whereas in the liver, many of those genes that were associated with traits are lipid traits. And that again kind of made sense to us. If the liver is involved in lipid regulation, whereas the islet might be more involved in diabetic associations. So that was a sort of a proof of concept that we can identify genes in the mice that show differential regulation as a function of strain, ask if the human homologue is associated with disease risk in GWAS, and see this kind of enrichment for diabetes or lipid-related traits. And again, I just want to show you a few examples of them. So in islet, H-hex is a key transcription factor associated with delta cells, and delta cells make somatostatin, a very important hormone that is secreted by the islets. And you can see in the islet, there are dramatic differences in H-hex expression, and that might suggest that there are differences in delta cell complement and islets among these mice. And in the right, I'm showing you the association plot indicating that H-hex is very strongly associated with diabetes in humans. Here's a gene, PPAR gamma, a very important nuclear receptor. And it turns out that in the liver, PPAR gamma is showing a relatively low expression in these 3 wild-derived strains. And PPAR gamma, probably not surprising to anybody, is strongly associated, and these huge goers are very large with many traits. So that's interesting, and if you wanted to study PPAR gamma, some of these mice might offer an ideal model system to understand these relationships. Finally, just to tell you that almost every tissue, I can show examples. Here's one, a gene called MASP1, which is associated with the complement pathway and it shows dramatic reduction in expression in WSB mice in the heart, and MASP1 in humans is very strongly associated with a variety of cardiovascular related traits in humans. So I can show you many, many examples. There are thousands whereby we see dramatic differences in expression in the mice and the human homologue decorated with SNPs associated with traits, as I show you here for MASP1. So that sort of is starting in the mice and moving to the humans, but I want to do the reverse. That is, I want to ask, well, let's identify genes in the human that have a very strong association to one or more physiological traits, and then ask, well, what do they look like in the mice? And in order to do that, once again, I went back to Jason, and this has his fingerprints all over it, as you can tell. He provided me a list of human genes and their huge score for traits that I asked for that were linked to these categories. So anthropometric, which would include like body weight traits, hepatic, lipids, cardiovascular, glycemic traits, and type 1 diabetes. And you can see for each category, there are a number of gene trait associations that have very strong evidence, and a very strong evidence is a huge score of around 3.2. And so I really focused on genes whereby the evidence that that gene is associated with the trait was super strong. And I want to tell you a story where I basically focused on genes associated with type 1 diabetes in particular. And the top hit, or a top hit for type 1 diabetes in humans, is this gene called CTLA4, which is a negative regulator of T cells. And as I understand it, it goes up in expression as an attempt to compensate for the invasion of T cells as part of the autoimmunity in islets. And what I'm showing you here is that here is the expression of CTLA4 in islets from our founder mice. And what I'm highlighting is that there is a dramatic elevation in the expression of CTLA4 in islets from NOD mice. And let me say that that's not new. Basically, we've rediscovered what's already known, that NOD is a known model of type 1 diabetes, and that's consistent with this elevated expression of CTLA4 in those islets. And here's the regional plot, the PHIWAS for CTLA4 in humans, showing that type 1 diabetes is a super strong hit for that association. It turns out that there are a lot of genes that are known to be associated with type 1 diabetes. And again, that comes from Jason's huge score for type 1 diabetes association in human GWAS. When I look at the mice, I see many examples of genes that are associated with type 1 diabetes and have elevated expression in NOD islets. And here I'm just showing you 6, but it turns out that there's many genes. And here's a heat map now where I'm depicting the expression across all of the mice. And I think you can appreciate for the NOD mice, which are shown right here, there's an increase in the expression of all of these genes associated with type 1 diabetes. Again, confirming what we knew is that NOD offers a model for that disease. But what we didn't know, and it was striking, were the number of genes shown in the bottom panel now that are also have a striking elevation in NOD that are not known to be associated with type 1 diabetes. And most certainly, many of these genes are not islet genes. They're probably associated with the infiltration of the lymphocytes as part of the autoimmunity. But I reached out to our colleague, Clayton Matthews, who's an expert in type 1 diabetes, and worked with Ed Leiter, developing the NOD as a model of type 1 diabetes. And I'm going to work with him to try to tease apart which of these genes might offer new markers of type 1 diabetes, not known to be associated with, but perhaps some actually in the beta cell and playing a role in the etiology of type 1 diabetes. And importantly, all of that I just described to you was first spawned by taking these genes provided to me by Jason as being associated with type 1 diabetes. And that same kind of query could be done for any of these genes associated with other traits, and then asking if they have differential regulation in the mice as a function of tissue type. So I want to switch now and talk about the second study where we went back to the founders, and here we challenged them with 2 different diets. And that's linked to Alan's earlier comment about looking at the genetics of diet responsivity. So we put the mice on either a high carb or a high fat diet for 18 weeks, and then we did a number of measurements, including RNA-seq, lipomics in collaboration with Will Holland, and metabolomics in collaboration with Dick Kibbe. And I'm going to show you a few striking examples of that survey in liver. So it turns out SCD1, a well-known gene involved in lipogenesis, is the most diet responsive gene in liver. And I'm showing you in this particular plot now, developed with a shiny tool made by Brian Yandel, in that in every single strain, for whether the mice were the female or the male, and I'm just showing you AJ over here, there's a dramatic reduction in the expression of SCD1 across the board. And that's consistent with SCD1 being very, very diet responsive. But contrast that to this gene, phospholipid transfer protein, PLTP, where there's a diet-dependent reduction in most strains, but not all. And I'm highlighting CAST here, and CAST is one of the outliers for a lot of the phenotypes that we look at. But whereas diet causes a dramatic reduction of PLTP in, say, NZO, it actually causes an increase in the expression of PLTP in CAST. And that's very interesting because it suggests that strain background is influencing the diet-dependent regulation of this gene. And the reason that's interesting to us is because PLTP is very strongly associated with lipids in humans. And so our data would suggest that the diet-dependent regulation of this gene that is different as a function of genetic background might contribute to the association of lipids in humans. Like I said, in collaboration with Will Holland, we've done lipomics. And just like we see gene expression in the liver, we have a number of lipids, and here I'm showing you liver triglycerides, again using Brian Yandel's shiny tool to indicate that here's a case of a particular lipid that is associated with NASH in humans, where it goes up as a function of diet in most, but not all strains. So you can see a dramatic increase in these strains, whereas in the wild-derived strains, and in particular CAST, there's actually a decrease in liver triglycerides in response to the same diet that in some strains causes a dramatic increase in liver triglycerides. And it's that kind of interplay between genetics and diet responsivity that we're working very hard to understand now, and its contribution to association in human GWAS. Finally, I want to end with 2 more stories because Alan talked about the diversity outbred mice, which are the progeny of taking these 8 founders and shuffling their genomes. And when you do that, you create these amazing mice that are all genetically distinct. They have tremendous phenotypic diversity, and these are more representative of the kind of genetic mosaic nature that exist in humans. They're all genetically distinct. They're not 95% heterozygous at all loci, and they have huge phenotypic diversity. And they offer an ideal platform for us to start asking genetic questions, because everything that I've talked about so far has been a genomic question. That is, CAST is different than B6. But now with these mice, when we make those same measurements, we can do genetic association mapping. And I'm going to show you 2 examples that are striking, of which there are hundreds or thousands of these cases. So here's the expression of PNPLA3, a key triglyceride lipase in the liver. And what I'm plotting here is the genetic evidence that there's regulation of this gene. And you can see in the top panel is when we put the mice on a high carb diet, there is no evidence of genetic regulation of PNPLA3, whereas for the mice that were on the high fat diet, there's a very strong cis EQTL. And what that means is that there's local genetic differences that result in a change in the expression of PNPLA3, but that it only manifests depending on diet. And this really illustrates the genetic of diet responsivity, at least for the regulation of this key triglyceride lipase in liver. What's interesting is if you go back to the founder mice and you ask, well, what is the expression of PNPLA3 doing as a function of strain, sex, and diet? Once again, we see dramatic diet-dependent regulation in some, but not all mice. And again, CAST and WSB appear to be outliers compared to NZO. And as you might expect, PNPLA3 has very strong association to various traits in human GWAS. So with the advent now of the DO mice, or the diversity outbred, we can start to ask these genetic questions and what part of the regulation of the genes or these other traits like lipids and metabolites show diet dependence for that genetic regulation. And I'm going to show you one more example because it illustrates something that is really powerful, that we're able to identify not only cis EQTL, and here's a gene, a lysosomal enzyme, that has a very strong cis EQTL regardless of diet, because it's physically located at this locus on chromosome 8. But here is what is called a trans EQTL. So the gene is physically located on chromosome 8, but its expression maps to this locus on chromosome 5. And that's very interesting because it implies that there is some key regulatory factor at this locus on 5 that is influencing the expression of this gene. But it's even more interesting that we see it manifest only for the mice on a high carb diet. So we're in the process of cataloging all of these EQTL, or expression quantitative trait loci, that are diet dependent. And some of them are not going to be diet dependent, as I show you here. Some of the trans EQTL will be diet dependent, and some of the cis EQTL, as I showed you a moment ago, are also diet dependent. And we're using that information to get at the genetics of diet responsivity, at least for hepatic gene regulation. Like I showed you with PNPLA3, this gene GAL-NS is also associated with various traits, including fasting insulin. And that is the association at the GAL-NS locus. But because, let me back up one slide, our data suggests that there is a locus on chromosome 5 that plays a key role in regulating GAL-NS expression. I asked the question about, well, what about that region to which GAL-NS maps in trans? So now what I'm looking at is a regional association plot from GWAS, and you can see many, many traits, including lipids, that are associated to the region that is synthetic to that locus that GAL-NS maps in trans. And we're beginning to ask those questions as well, integrating our mouse data with human data.
Hey, Mark, a quick question from Nora in the chat. She
Yeah.
asked, what is the comparison diet for the experiments? This is, I think, from another slide beforehand.
It's a super high fat, low carbohydrate, or it's a high carbohydrate, super low fat diet. That's what we're forcing the mice to consume.
Beautiful. And then Samir has his hand raised. You're welcome to unmute yourself. You have a question?
I have one more slide.
Yeah. Okay. Then we'll wait.
Okay, and
Go
then,
for it. Hold on.
and I've been monitoring, so we've got 15 minutes left.
Yep, I think we're
So
good.
we're
Yeah.
in the process of constructing a user-friendly web portal that would basically give everybody access to this data. And it's under construction, so consider this a beta, a pre-beta version. But it will allow the user to type in their favorite gene and look at its expression in the 5 tissues for which we've profiled. And right now we're importing the data from that study number 1, where the mice were on one diet. So we're looking at the influence of strain background and sex on gene regulation in any of these tissues. It will quickly generate a plot. And let me just say that this plot's going to change its appearance in terms of the mice are in the wrong order and they have the wrong colors. So this is, again, just a beta version, but it will quickly generate a plot. It will also list all of the genes that you can order by their influence by strain background. And then it will immediately generate a regional or a PheWAS plot showing how that human homologue is associated with one or more traits in humans. And it began with entering the gene, looking for an expression profile. All of these buttons here will be hyperlinked. So if you click on one of these, it will repopulate this plot for the expression in the particular tissue in mice, and will also repopulate the PheWAS. And we're going to add a toggle here where we can have just the genes for which there's a human homologue listed, or all of the mouse genes. And many of these guys, these GMs, they show dramatic regulation as a function of strain background, but they may not have a human homologue, but could still be very interesting. Finally, let me just say that I love doing this work, as you can tell by my passion. And while we build this thing, this is an open invitation. If anybody has a favorite gene and they want to know what it looks like, please email me and I'd be happy to look and give you a gene report. And then it's my privilege to present all of this. And of course, this is team science. And I basically, everyone listed here plays a key role in these measurements, and it's been my honor to present it. Thank you.
Excellent. Thank you, Mark. Let's open the conversation up for questions, and then Samir, you've had your hand raised. Why don't we start with you?
Okay, great. I had a question about the maybe second to last data piece where you asked a really interesting question about whether variants that are in the region that's a trans EQTL for that gene. Yeah, this data. So
Yep.
A question about what's specifically being done here. Did you pick, is this a one SNP, multiple SNPs in the region?
No, that's a great question. So I went over that quickly, but it's really important. So here, this is the physical location of this gene. And so when you go to the type 2 knowledge portal right now and type in your gene and hit gene-centric view, it'll bring up this, right? So this is the huge score that the gene is associated with these traits.
Okay.
Whereas our data, if I back up, says that there's a key regulatory locus right here on chromosome 5 that is affecting the expression of GAL-NS. So what I wanted to know is, well, what about this region? And so I identified this region, which was about 2 megabases, and I did a liftover to identify this syntenic region in the human.
Uh-huh.
And most likely it's not on chromosome 5. I forgot where it is. But what I did is I identified the region that is syntenic to this trans EQTL. And then I did a regional association plot to develop this picture here.
Okay. So in other words, multiple SNPs are being evaluated here.
For sure. And,
Okay.
and it's big. So I didn't even list a gene here because there's probably a lot of genes under this locus. But it starts to imply that not only do we have a gene-centric view that we're interested in, but we want to identify the trans loci. And I can tell you that there are thousands of trans EQTL for these genes that are diet dependent. And we're very excited by that. And one way that we sort of prioritize those is ask, are there regions to which there are a lot of traits that co-map? And the answer is yes. There are hotspots of trans EQTL and that are diet dependent. And we're very carefully looking at them to see, well, what gene is sitting there? And moreover, what traits in humans are associated with that syntenic locus?
Got it. Thank you.
Sorry, I was muted. Another question in the chat is the link available for the portal? And the answer to that is eventually, and it will be. what the way that we build these typically is we start with a portal that's sort of outside of our main portal, but it's eventually going to be open access, but it's built in the same framework, and the data are integrated in the same way that we build our entire platform around, which is really nice because once we want to integrate it fully, it's there. So we want to offer the opportunity to sort of test it, really tweak it, get feedback, allow you to say the things you just said to me, or like, what if I want to do this, or how do I do that, and sort of help us think about how you would actually use it, and then we'll get it integrated into the portal. So that's kind of the story that, they're in the process. But really, moreover, what we're hoping is this is really a foray into, making it more straightforward and easy for a person who's searching their favorite gene to say, if I know about in the human, what's going on in the mice, and see that sort of seamlessly. So we have some infrastructure already planned with Mark and Alan to build this prototype, but also we have to do this more broadly, right, across all common diseases for all of our portals. So this is just the beginning of a conversation. I'm really curious if folks on this webinar or others have ideas. We really want to hear them, because this is sort of exactly how we do this, right? So this is so you're seeing how it's all made, but also like, how does this actually become something that is more applicable and scalable across the entire portal universe?
We have another question in the chat. Are the diversity outbred mice you are using the same as the collaborative cross mice?
That's an excellent question. So there's the CC, or the collaborative cross. Those are recombinant inbred lines, and they're distinct from the DO mice, which are the diversity outbred. And the main distinction is that the diversity outbred were generated by outcrossing the CC mice. And unlike the DO, which are heterozygous at most loci, the CC lines, of which I think there's on the order of 100 or so, they're homozygote at all loci. They're still genetic mosaics across their genome of those 8 founder strains, but their genome structures are different in terms of homozygosity versus heterozygosity. And we haven't used the CC mice for our studies. We've relied more on the DO mice, in part because of their heterozygous nature, approximates more closely what is true for humans in terms of their genome structures.
Other questions, comments from folks? And then I should mention, I put this in the chat, that the results that Mark was sharing from Jason are from a new version, a very cool upgrade from the huge score, which we call Pigeon. And it's a fun acronym. But really, it doesn't just incorporate human genetics, it incorporates model systems, incorporates pathways. It's really quite lovely. And that is in a beta version also available. And that's where Mark got these results. So if anyone's interested in seeing some results there, please feel free to email us directly, and we can actually look on what our huge scores now are updated to for your favorite trait. But that will be available live on the portal, hopefully in October. So all those results will all be incorporated by then. And I'm hoping maybe by October, we'll also have a lot of progress on this mouse resource as well, to pull that into the overall CMDKP.
And let me say also, Noelle, that right now, what we're building this out with the data from what I called study number 1.
Right.
Which were the 8 founders on one diet, and really we're looking at the influence of strain and sex, of which there's a lot. Study number 2 is also available, and ultimately we want to get this in here, which then asks the diet question, and that's all founder data. Ultimately, we want to get the diversity outbred data,
Right.
that allows you to ask the genetic association question that I showed you for just 2 genes, PNPLA3 and GALNS. That data is super powerful because it really identifies regions that explain why the founder mice may be different for certain traits.
That's exactly right. Another question from Mark Herman. Have you incorporated metabolomics data yet?
Great question, Mark. We actually are in the process of doing that. I had planned to show some metabolite data that we are collecting in collaboration with Dick Hibby at Yale. And I just felt like I didn't have time to rush through that. It's super interesting. With Dick, his mass spec platform is able to measure more than 1,000 molecules. And we're doing that in the plasma and the liver of the DO mice. so that we can again ask the genetic question for mapping these molecules. We have founder metabolomic data from Dick as well. And I focused on the RNA and the lipid data, but we also have that data that ultimately we want to incorporate into this tool as well.
Beautiful. Other questions? Well, good news, you guys finished beautiful timing. These are great conversations. And also, if folks want to see this, we put it on YouTube, we post it and share it. We'll also share the links to the beta version of the portal when they're available. But if you have any questions, please email help@kp4cd.org. It goes to literally me and a couple other people. We get back to you within 24 hours, or on a specific gene, talk to Mark directly, because I think that's really something that I think a lot of you will be interested in. So copy down Mark's email if you're curious about.
Well, let me say, I'm selfishly doing this because I actually love to give people presents. So please don't hesitate to reach out to me while we build this thing. And I would be happy to provide you the data.
Oh, Ashley raised her hand. Ashley, would you like to unmute yourself? There you are. Hi.
Thank you. Thank you. Thank you. Wonderful presentation. I have a quick question in terms of whether you actually exercise your mice.
No, we did not. I mean, it's an interesting question, Ashley, because you certainly could do an exercise study. And I believe that some people have actually asked about longevity of treadmill as a function of genetic background. And some of the mice, I think, fall off the treadmill very quickly, and others just run and run and run, very similar to what has been seen in rats. But you could do a genetic study and ask, are there loci that confer longevity of exercise if done in the context of the DO mice? But we have not pursued those studies.
Yeah, because we all know diet and exercise is a kind of a first line intervention for diabetes. So
Yes.
thank you.
Beautiful. Well, with that, I want to really thank Alan. Thank Alan for his inspiration multiple years ago that's now kind of finally coming to fruition. And thank Mark for sharing his results and doing these analyses and helping us build a new resource that will hopefully make the CMDKP more useful to a broader community in the future. I really appreciate that.
And thank you, Noel, to you and your team. It's been a real joy to work with you, and we look forward to a lot more very productive work together.
So do we. Fabulous. All right.
Yes.
Thank
Let
you,
me
everyone.
echo. Thank you, Noel. Thank you, everybody.
Thanks,
Bye
everyone.
now.
Have a great August. Take care. Bye-bye.
Bye.